Swayam Prabha. Digital India. Educated India.
Hello learners, I am Dr. Rekha Sharma, Professor, UGC Human Resource Development Center, Rashtrasanta Tukaroji Maharaj, Nagpur University, Nagpur. In this module, we shall study Minerals Part 2, wherein we will be studying about iron and iodine. Now objectives. After studying this module, the learners will be able to describe the functions and food sources of iron and iodine, discuss the various deficiency and toxicity symptoms of iron and iodine, and identify the dietary requirements of iron and iodine. Now let us start with iron. The total iron content of the normal adult man of 70 kg weight is estimated to be about 4 to 5 grams. A greater part of the iron in the body is present as hemoglobin. Most of the body iron exists in complex forms bound to protein either as porphyrin or heme compounds or as ferritin and transferrin. Free inorganic iron occurs in the body only in very small amounts. The hemoprotein and flavoprotein enzymes also contain iron. Now let us study hemoglobin. The major function of hemoglobin is the transport of oxygen from lungs to all the tissues of the body. The oxygen binding capacity of hemoglobin is 1.34 ml oxygen per gram. The oxygen bound hemoglobin is referred to as oxyhemoglobin. Hemoglobin also transports carbon dioxide from tissues to lungs. The carbon dioxide bound hemoglobin is referred to as carbaminohemoglobin. Now let us study functions of iron. Iron is an essential part of several oxidative enzymes. It helps in specific brain functions such as good attention span and capacity to learn and memorize. It facilitates the complete oxidation of carbohydrates, proteins and fats within the cell and release of energy for performing physical work. Now let us study the dietary sources of iron. The human diet contains two types of dietary iron, heme iron and non-heme iron. Heme iron is present mainly in hemoglobin and myoglobin present in meat, fish and other animal foods. Heme iron derived from animal food is absorbed directly in the human gut to the extent of 60 to 70%. Non-heme iron is obtained from cereals, legumes, pulses, fruits and vegetables. The non-heme iron would get absorbed in the body easily by including vitamin C rich foods. Ascorbic acid probably forms a chelate with inorganic iron that remains soluble at the alkaline pH of the duodenum. Now cereals are the most important source of iron in the diets of a large majority of the population in India and other developing countries. Other important sources are legumes, green leafy vegetables and jaggery. Milk is a poor source of iron. Now let us study factors affecting absorption of iron. Role of the stomach. Now presence of acid in the gastric juice may help in solubilizing dietary iron. Absorption of iron is impaired in hypoacidity. Now second is ferrous versus ferric iron. Iron is absorbed in the ferrous state. Now, other factors affecting absorption of iron are: first is ascorbic acid. Now, absorption of iron is enhanced by the simultaneous administration of ascorbic acid. Now, phytic acid. The presence of phytates is deterrent to the absorption of metals, principally of calcium and iron. Now, sources of phytic acid are grains such as wheat, rice, oats, legumes such as soybean, lentils, peanut, nuts and seeds such as almonds, sesame seeds, tubers such as potatoes, beets and carrots. Now, oxalic acid. Oxalic acid present in certain vegetables forms insoluble iron oxalate and prevents the absorption of dietary iron. Examples of oxalic acid containing foods are spinach, beets and sweet potatoes. Then tea and coffee. Tea and coffee with meals decreases absorption of iron. Now, ferric iron or non-heme iron in the absence of protein and ascorbic acid are poorly absorbed. Now, we shall study iron deficiency. Effects of iron deficiency are cognitive impairment, decreased physical activity, and reduced immunity. Iron deficiency in school-going children leads to poor memory power, attention span, and poor scholastic achievement. Now, iron deficiency anemia. Iron deficiency anemia is widely prevalent among children, adolescent girls, and expectant and nursing mothers in all developing countries. The clinical features are the results of diminished oxygen carrying power of the blood due to low hemoglobin content, that may be 5 to 9 grams per 100 ml of blood. Now, iron deficiency anemia in women of childbearing age. The clinical features are general fatigue and lassitude, breathlessness on exertion, giddiness and pallor of the skin. In severe cases, there may be some edema of the ankles. Spoon-shaped nails is the common symptom of iron deficiency. The hemoglobin levels commonly range between 5 and 9 grams per 100 ml of blood, and RBC count, that is red blood cell count, to 3 to 4.5 million per cubic millimeter. Now, iron deficiency anemia in weaned infants and young children. Iron deficiency anemia is widely prevalent among weaned infants and young children in India and other developing countries. The hemoglobin levels in severe and moderately severe anemic cases may range from 5 to 9 grams per 100 ml. The children are dull and inactive and show pallor of the skin. The appetite is poor and growth and development of children are retarded due to low food intake. There is tendency for children below 3 years to eat mud. Now, after studying iron deficiency anemia, we shall now study iron toxicity. The absorption of iron is very effectively regulated. This prevents overload of the tissues with iron from diet or supplements in normal healthy individuals. However, an excessive body burden of iron can be produced by greater than normal absorption from the alimentary canal, by parenteral injection, or by combination of both. For instance, people with genetic defects develop iron overload as it occurs in idiopathic hemochromatosis. Idiopathic hemochromatosis is a hereditary disorder of iron metabolism characterized by abnormally high iron absorption owing to a failure of the iron absorption control mechanism at the intestinal level. High deposits of iron in the liver and the heart can lead to cirrhosis, hepatocellular cancer, congestive heart failure, and eventual death. African or Bantu siderosis, chronic liver disease, pancreatic insufficiency, shunt hemochromatosis, and certain types of refractory anemia have been found to be associated with iron overload. It has recently been shown that excess iron intake via overuse of iron supplements could pose a possible health risk. Cellular and tissue injury due to free radical reactions appears to be the possible mechanism. Normally, iron is bound tightly to the proteins. Now, requirements of iron. Iron requirements of persons consuming a predominantly cereal based diet will be greater than those consuming large quantities of meat and eggs. Iron requirements suggested by Indian Council of Medical Research 2020 are 19 milligrams per day for adult males and 29 milligrams per day for females. For pregnant and lactating mothers, the iron requirements are 27 and 23 milligrams per day respectively. Now, iron fortification. Food fortification is the process of enriching food with essential vitamins and minerals that regular diets often lack. Food Safety and Standards Authority of India, that is FSSAI, under Food Safety Standards Fortification of Foods Regulation 2016, has recommended the fortification of staple foods like rice and wheat flour with iron. Under this regulation, rice and wheat flour are being fortified with iron, folic acid and vitamin B12. Now let us see how much amount of this iron is fortified in rice. As I said, rice is being fortified with iron, folic acid and vitamin B12. Now FSSAI, that is Food Safety and Standards Authority of India, has recommended addition of ferric pyrophosphate 28 milligrams to 42.5 milligrams per kg or sodium ferritate 14 milligrams to 21.25 milligrams per kg in rice. Now, iron fortification in wheat flour. Like rice, wheat flour is being fortified with iron, folic acid and vitamin B12. Food Safety and Standards Authority of India has recommended addition of ferrous citrate or ferrous lactate or ferrous sulfate or ferric pyrophosphate or electrolytic iron or ferrous fumarate or ferrous bisglycinate 28 milligrams to 42.5 milligrams or sodium ferritate 14 milligrams to 21.25 milligrams per kg in wheat flour. Now, iodine. Iodine is an essential micronutrient required for the normal mental and physical growth and development. The thyroid hormones are indispensable for normal growth and development in humans and animals. Synthesis of iodine containing thyroid hormones occurs exclusively in the thyroid gland. Iodine as an element was discovered only in 1811. However, its presence in the thyroid gland was discovered by Baumann et al. In 1895. Now, functions of iodine. Iodine is an essential constituent of the thyroid hormone, thyroxine, that is T4, and triiodothyronine, that is T3, which have a key role in growth and development. Most of the iodine in an adult body is found in the thyroid gland. The only known function of iodine is as a constituent of thyroxine. The thyroid hormone regulates the rate of oxidation within the cells. The iodine absorbed is incorporated into the amino acid tyrosine to form the hormone thyroxine. If intake of iodine is inadequate, the stores of thyroxine are gradually depleted and the thyroid gland enlarges in an attempt to produce the necessary thyroxine. Now let us study the sources of iodine. The iodine concentration in foods is highly variable and also depends on the concentration of iodine content of soil in that region. The iodine present in the upper crust of the earth is leached by glaciation and repeated flooding and is carried to the sea. Hence, sea water is a rich source of iodine. The seaweed located near coral reefs has an inherent biological capacity to concentrate iodine from the sea. The iodine content of drinking water is a valuable index of the iodine content of the rocks and soils of a region and hence of the locally grown foods and has been correlated with the incidence of goiter in many areas. In addition to water, iodine is also contributed by seafoods. However, a large difference in the content exists between seawater fish and freshwater fish. Sea fish contain about 300 to 3000 micrograms of iodine per kg in contrast to 20 to 40 microgram iodine per kg in freshwater fish. The examples of seawater fish are sardines, mackerel, ribbon fish, grouper, cobia, and tuna. Examples of freshwater fish are mahseer, trout, carps, catfish, snakeheads, and featherbacks. The iodine content of foods of marine origin is much higher than that of any other class of foodstuff. That differences among the latter becomes relatively insignificant. The iodine content of other foods in micrograms per kg on the fresh edible basis are fresh fruits 20, leafy and root vegetables 30, cereal grains 40, meats 40, milk and milk products 40, and hence eggs 90 micrograms per kg of fresh edible basis. Food additives used as bread dough oxidizers or conditioners can contribute to the iodine content of the diet. Now, iodine deficiency disorders. The term iodine deficiency disorders, that is IDD, includes a spectrum of disabling conditions affecting the health of human beings starting from fetal life through adulthood, resulting from inadequate dietary intake of iodine. Iodine is required in our body for the synthesis of thyroxine, the hormone produced by the thyroid gland. When iodine intake falls below the recommended levels, the thyroid gland is no longer able to synthesize hormone. Every year, 9 million pregnant women and 8 million newborns are at risk of iodine deficiency disorders in India. In India, due to lack of iodine in the soil and therefore in the diet, all 1.2 billion people are at risk of iodine deficiency disorders and around 264 million people are at high risk. Iodine deficiency disorders are linked to iodine deficient soil due to glaciations, flooding, river changing course, and deforestation. The iodine present in the topsoil is constantly leached. This in turn leads to deficiency of iodine in crops grown on iodine deficient soil, with consequently low iodine in the diet for livestock and humans. Iodine deficiency affects all populations at all stages of life, from the intrauterine stage to old age. However, pregnant women, lactating women, women of reproductive age, and children younger than 3 years of age are considered the most important groups to diagnose and treat iodine deficiency, because iodine deficiency occurring during fetal and neonatal stage leads to irreversible damage to the brain and central nervous system and consequently to irreversible mental retardation. Iodine deficiency disorders comprise of a range of disorders including goiter, hypothyroidism, cretinism, brain damage, intellectual disability, psychomotor defects, hearing and speech impairment, abortion, and stillbirths. Intelligent quotient, that is IQ, children born in iodine deficient areas have 13.5 IQ points less than those in iodine sufficient areas. A majority of the consequences of iodine deficiency disorders are invisible and irreversible, but at the same time are totally preventable. Iodine deficiency disorders constitute the single largest cause of preventable brain damage worldwide. Now, goiter. One of the well recognized features of iodine deficiency disorders is goiter. If sufficient iodine is not taken in the diet, enlargement of the thyroid gland takes place, resulting in the disease called goiter. The thyroid gland of the adult, which normally weighs about 25 grams, may weigh as much as 200 to 500 grams or even more in goiter. If treatment with iodine is started very early, the thyroid may become normal. But if treatment is delayed, the enlargement of the gland persists. Now, hypothyroidism. Hypothyroidism is the principal factor responsible for the damage done to the developing brain and the other harmful effects. Hypothyroidism is accompanied by low basal metabolic rate, apathy, slow reflex relaxation time with slow movements, cold intolerance, and myxedema. Now, endemic cretinism. Cretinism is the result of iodine deficiency during pregnancy, which adversely affects fetal thyroid function. It is characterized by severe irreversible growth failure, mental retardation, deaf mutism, and spastic paralysis of legs. Neurological cretinism is characterized by poor cognitive ability, deaf mutism, speech defects, and proximal neuromotor rigidity. It is much more preventable than myxedematous cretinism, which includes hypothyroidism with dwarfism. Now let us study toxicity of iodine. Iodine-induced thyrotoxicosis or hyperthyroidism and toxic nodular goiter may result from excess iodine exposure in these individuals. Hyperthyroidism is largely confined to those over 40 years of age, and symptoms are rapid heart rate, trembling, excessive sweating, lack of sleep, and loss of weight and strength. Now, individuals who are sensitive to iodine usually have mild skin symptoms. Now, after studying goiter, we must also study goitrogens. What are these goitrogens? Goitrogens are substances that interfere with iodine metabolism in any way that inhibits thyroid hormone synthesis. These active goitrogens are released by plant enzymes from thioglucosides or cyanogenic glucosides found in cassava, cabbage, broccoli, turnips, rapeseeds, red skin of peanuts, and mustards. Most important of these is cassava, which can be detoxified by soaking in water and cooking it well. Tobacco smoke also contributes thiocyanate. Now, iodine requirements. Iodine requirements suggested by Indian Council of Medical Research 2020 are 140 micrograms per day for adult males and females. For pregnant and lactating mothers, the iodine requirements are 220 and 280 micrograms per day respectively. Now, prevention of iodine deficiency disorders. The Food Safety and Standards Authority of India, that is FSSAI, under the Food Safety Standards Fortification of Foods Regulation 2016, has recommended a double fortified salt for the prevention of iodine deficiency disorders and iron deficiency anemia. Double fortified salt, that is DFS, is an innovative new fortified food product delivering small but crucial amounts of iodine and iron to human beings through their diet. In general, DFS formulations are intended to provide 100% of daily dietary iodine requirement and about 30 to 60% of daily dietary iron requirement. Dual fortification of salt with iodine and iron could be a sustainable approach to combat iodine and iron deficiencies. As per the Food Safety Standards, Fortification of Foods, Regulation 2016, salt shall be fortified with iodine and may also be fortified with iron in combination with iodine at the level given below. Now, iodine content at manufacture level will be 20 to 30 ppm on dry weight basis, and at distribution channel including retail level it should contain 15 to 30 ppm, that is parts per million on dry weight basis, and the source of nutrient will be potassium iodate. For iron content, the level of nutrients will be, level of iron will be 850 to 1100 parts per million, and the source of nutrient will be ferrous sulfate or ferrous fumarate. Now let us summarize. The total iron content of the normal adult man of 70 kg weight is estimated to be about 4 to 5 grams. A greater part of the iron in the body is present as hemoglobin. The major function of hemoglobin is the transport of oxygen from lungs to all the tissues of the body. Iron is found in foods in one of the two forms, that is heme or non-heme. In human diet, the primary sources of heme iron are the hemoglobin and myoglobin from consumption of meat, poultry and fish, whereas non-heme iron is obtained from cereals, pulses, legumes, fruits and vegetables. Deficiency of iron causes iron deficiency anemia, cognitive impairment, decreased physical activity and reduced immunity. Iodine is an essential micronutrient required for the normal mental and physical growth and development. Iodine is an essential constituent of the thyroid hormones, thyroxine, that is T4, and triiodothyronine, that is T3, which have a key role in growth and development. The iodine content of foods of marine origin is much higher than that of any other class of foodstuff. The term iodine deficiency disorders or IDD include a spectrum of disabling conditions affecting the health of human beings starting from fetal life through adulthood, resulting from inadequate dietary intake of iodine. The iodine concentration in foods is highly variable and also depends on the concentration of iodine content of soil in that region. The Food Safety and Standards Authority of India, that is FSSAI, under the Food Safety Standards Fortification of Foods Regulation 2016, recommended a double fortified salt for the prevention of iodine deficiency disorders and iron deficiency anemia. These are the references. With this, we have come to an end of the lecture on minerals, part 2. Thank you.